which carries oxygen in your blood. Insulin regulates the amount of glucose or sugar in your blood. The coronavirus spike protein, this one's not to scale, helps coronavirus invade your cells through the ACE2 receptors, which is another protein. And the fun one is that luciferase makes firefly buds glow. And all of this diversity in function arises from 20 fairly simple building blocks called amino acids. We call them amino acids because on one end they have an amine group, and on the other end they have a carboxylic acid group, so together it's an amino acid. Okay. And then what makes each of the amino acids different from each other is the side chain, which in chemistry people use R to denote side chains that are variable. And there's approximately 20 side chains that occur commonly in natural proteins. From the simplest, which is just the hydrogen, you can get some more complicated ones. I think there's a CH3 and a COH, and so forth, with different kinds of chemical properties. And these chemical and physical properties give rise to the divergent properties between different amino acids, which give rise, and the combinations of those in a chain give rise to all the different functions that occur in nature. My interest is in designing proteins with new and useful functions. So some reasons you might want to do that. You might want to do new chemistry. Here is an example of a catalytic reaction that forms carbon-silicon bonds. And this reaction is known in organic chemistry, but until about 2017, it was not known to be done by any enzymatic routes through proteins. And the reason people are interested in doing this in general is that enzymes give you very precise control over the products you get out of reaction, and they're biodegradable. So you're not stuck with, the alternative is usually these heavy metal catalysts, which are very effective, but they're not as selective, and they are either expensive or toxic, depending on which heavy metals we're using. We can use proteins as therapeutics. There's many antibody therapeutics now, which are an example of antibodies are a class of proteins. I personally regularly use an antibody therapeutic for eczema. So unfortunately it's injectable, which is kind of inconvenient, but it's better than many of the alternatives. This molecule is called asparaginase. It's an enzyme that breaks down the amino acid asparagine. It's used as a chemotherapy agent. And it's used as that because it turns out that there are many tumors that can't make their own asparagine. So if you break down the asparagine in the tumor environment, it kills off the tumor cells, but your healthy cells can make their own asparagine. We also want to use proteins as molecular tools. The example here is Cas9, which is part of the bacterial immune system against phage infection. Phages are viruses that infect bacteria. And we have repurposed this to do gene editing in humans and other systems. So the general idea here is that in nature, we can observe many examples of natural sequences of proteins. And the sequence here is a chain of amino acids, the order and the composition of that chain. And that sequence encodes a three-dimensional structure for the molecule. And that three-dimensional structure, or an ensemble of structures, mediates some function in the cell or in the environment. The idea is to learn from that and do generative design so that we have some model that can generate new examples of proteins and hopefully do new and useful functions. And so here's that sequence structure function paradigm again. And there's an older protein design paradigm that basically says if structure is the thing that mediates function, if we can come up with a way to design a structure that will do the function we want, and then find a sequence that will fold to that structure, then my problem is solved. And so the challenge here is to create new, diverse, novel, and realistic and designable structures. So designable here means that I can find a sequence that folds into that structure. So this project was originally an intern project in summer 2022 with Kevin Wu, and it's now a paper out in Nature Communications. So to understand this project, we're going to dive a little bit more into protein biochemistry, not too much chemistry, I promise. I know most of you usually work a level below that. So here's amino acid. So to make that polypeptide chain, what actually happens is called a condensation reaction. So the OH from one residue, the N-terminal residue, and one of the amine hydrogens from the C-terminal residue leave and make water, and you get a polypeptide bond between the C and the N, carbon and nitrogen, from one to the next. And you can also see that the side chains kind of flip opposite directions. So let's take a look at this chain. And there's a nomenclature for the carbons here. So the carbon that's attached to the side chain, we call C alpha. The carbon that's attached to the oxygen is C. And we can actually determine protein structure with the coordinates of every atom, yes. But we can come up with a slightly more efficient parameterization by looking at these bond angles. So there's three bond angle rotational angles here. And there's these three diagonal angles here. And if you know what these angles are for every pair of consecutive amino acids in your chain, that fully defines the three-dimensional structure of the protein up to the side, up to the side chains. So if you also get the side chain rotation and the side chain configuration, then you have the full structure, but you actually can't design the protein with the side chains. So what we design is what we call the backbone, which is everything but the side chains. And the reason for that is if you know the side chains, then you know what the amino acids are. But if you know what the amino acids are, then you wouldn't be doing protein design, right? So we want to specify the backbone structure to be more precise, then go back and figure out what the side chains should be, or see if it's possible to find side chains that will fold in that structure. And so we call this model folding diffusion or folding diff. And the reason we do that is because we do the diffusion on those angles, on that angle representation. And in the diffusion model, you start with X0. In the forward model, you corrupt those angles to random angles. And then you, learn a reverse process parameterized by neural network that undoes corruptions. And then you start from random angles, sample from the normal distribution, a rapid normal distribution, and come up with new angles. And then those new angles hopefully encode a realistic three-dimensional conformation for the protein. Train this on about 30,000 structures from CAP. These are fairly small structures. This is 2022, and this is fairly new. People can do much bigger structures now. And the one advantage of diffusing on the angle instead of directly on the atomic coordinates is you can just use a vanilla transformer without having to encode the equivariances into the neural network, because you can code it in the representation. Here's, I believe, an animation that might work. Generation. Right, so on the left, you see diffusion on coordinates. On the right, you see diffusion on the angles. And it's kind of motivated by the physical folding process, but it doesn't actually try to replicate. So we're going to evaluate these generations at three different levels. We're going to look at how realistic are the generated angles. Then we look at, do we generate reasonable looking motifs? And then the overall structures. So first thing we do is we generate a bunch of angles, and then we look at the distribution of these angles and match them to the distribution of the test. And you can see that for all 6 angles we consider, the generated distributions match the natural distributions for each individual angle. But of course, these distributions are actually correlated. These are all marginal distributions. We actually care about the joint distribution. So let's see if we can capture some of those correlations. So we look at 2 specific angles, the phi psi angle, sorry, and plot those together. And we look at phi psi because they vary a fair amount, and because there are clear clusters that define what's called secondary structure in protein structure, which is sheets and helices and loops. So you can see that here is the real distribution from the test set. On the right is a generative distribution. And you can see we also generate different types of secondary structure, just like you would expect from the test set. So we generate SPI-side correlations that define common structural motifs in a way that is similar to natural recurrent proteins. And then we can also actually look at those secondary structures. So here we generate things, and then we use a different model to annotate. It's not a machine learning model. This is a deterministic model to determine whether every residue that we generate is part of a helix or a sheet. So here's examples of alpha helix, example beta sheet. These are your two most basic protein structures. And look at the counts for alpha sheets and beta helices in the test set versus the generated ones. And here you can see that we also look pretty close. And then we look at real structures from the generated structures from both the training set and test sets. And they kind of look reasonable. Some of them are a little more wobbly, more spaghetti-like than you'd like. And we look at these quantitatively. So we can measure this designability, which once again is, I've hallucinated this backbone structure. I don't know that it's actually possible to find anything that will make the structure. So let's see. I take following diff and generate something. We can run it through a different model that someone else has trained called protein at PNN, and what that does is it's train natural structures to, given the backbone, find the amino acid sequence that will fold to it. So that gives us a sequence. Then we can use a folding model that takes that sequence and predicts the structure and see how well those match. And we look at we're a measure called TM score, which goes from 0 to 1. Higher is better, and above 0.5, biologists consider that to be the same, what they call fold. So they're the same family of structures. And so if we look at the self-consistency TM scores, generally short ones are easier than long ones, as you can see. And if we look at this compared to an earlier model that works directly in the atomic level, so we do better on both the short and long ones. Nowadays, there are models that can do much better than this, using a lot of pre-training tricks and a lot of architecture tricks, and also much more data. We also check, and they are indeed better than random sets of angles, so the correlations do matter. If you just sample from the marginal distributions, your designability is much worse, which makes sense because the joint distribution does matter a lot here. Our generations are diverse. So folding diffusion is an example of kind of general model that is kind of the first step for generating functions, right? Because right now we're just generating structures that do something, or anything, or nothing. Ideally, eventually you want to specify the function and go directly to structure in the sequence. We generate protein backbones by mirroring the process, uses internal coordinate representation. We can generate realistic motifs and also realistic and diverse samples. If you want to run this code, it's on GitHub. You can look at the paper, and it is in Hugging Face. All right, I'll pause here for questions before we go to the second part. All right, keep going then. So, like I said, the traditional protein design paradigm is I have some function I want to do. I use some human knowledge or biological knowledge or physical knowledge to figure out a structure I think will do that function. Then I use some method, either a physical method or machine learning method, to design a sequence of amino acids that I think will fold in that structure. Then I go make it in the lab and hope it works. But, and this is kind of a viewpoint, is that sequence this universal protein design space. And what we really want to do is go directly from sequence to function. or from function to sequence, and kind of skip out that nuisance variable. And so we're going to do diffusion directly in sequence space. This is the preprint out now. This also started as an intern project, and then has grown a lot since then to involve a fair number of people from MSR. And we call this model EvoDiff, for evolutionary scale diffusion. So it's the same idea. In the forward diffusion process, we start with a sequence of amino acids. I'm using letters to represent the amino acids, because 20 amino acids, 26 letters is almost 1-to-1. And there's a few ways you could think of doing diffusion in this discrete space, and we tried a few. There's even more out there. One way is at every diffusion step, I'm going to corrupt by picking one uncorrupted location and replacing it with a mask token, which is a £. And if I have L amino acids in my sequence after L corruption steps, I have nothing but masks. And the train time, what that looks like is you take your sequence, you sample some positions to mask, and you predict what those locations were originally based on context from the unmasked locations. And if you do this for many sequences and many masks, you should learn a model that can learn to generate at every step. It can learn to unmask at every step. So we also try, and we call this OADM, which is order agnostic diffusion model. The original Hugo paper actually calls it an order agnostic autoregressive diffusion model, but we decided that was too long. So this one is called D3PM, which stands for Discrete Denoising Diffusion Probabilistic Model. And the similar idea, you start with real sequences from a data distribution. But now instead of replacing things with a mask token one by one, at every step during the forward corruption process, you use a transition matrix to probabilistically mutate every location. And then during training, the model is given the mutated sequence in the time step, and it tries to predict the original sequence. But generation time, you start from the sample from the uniform distribution and try to denoise it step by step to something that looks realistic. You can try different transition matrices here. We tried a uniform matrix. We tried this matrix derived from Blossom, which is where people took sets of evolutionarily related proteins and solve what mutations were common, which ones were uncommon, and you upweight the common ones and downweight the uncommon ones. But we had really high hopes for this like evolution-inspired transition matrix. Unfortunately, it turns out that no matter what transition matrix you use in D3PM, it does worse than OADM. So we are going to use that one moving forward. We call this EvoDiff-seq because it operates in sequences. And the explanation behind what we call evolutionary scale is because we're training on 15 ON sequences taken from across the scale of evolution, meaning they're from many different organisms and they perform many different functions. And this is parameterized as a 640 million parameter CNN. The generation time, what this looks like is you start with all mask tokens and you sample a decoding order and infer what those residues should be one by one. On the right is just a structure prediction for every intermediate generation. It's not actually like how the protein folds or anything, just pretty pictures. And so in this case, we generate a sequence and we use a different model to predict the structure for that sequence to visualize it. And EvoDiff-seq generalizes previous mask and autoregressive language models in the sense that we're learning every step of every possible decoding order, right? So the model learns to generate from all masks to full sequences, and it learns every possible decoding order given infinite training time. In the traditional left to right autoregressive model, you're given a start token and you predict the next token one at a time. This is how things like GPT work. You give everything to the left, the model predicts the next token to the right until the end. So this learns one decoding order. right? The left to right decoding order, which is actually an easier task because given the same model capacity, you only have to store one decoding order. The reason we want to do multiple decoding orders is we want to do conditional generation, where we condition on part of the sequence. We fix part of the sequence that we know we want, and want to generate around it, which you can't do in a straightforward way with these left to right models. There's also another line of work in languages called BERT. In general, I refer to this as what's called a mask language model. And these models, they mask a 6% of the sequence. They also mutate a small percent of the sequence and predict the original sequence. And this was originally designed to be a pre-training task and then used downstream to predict structures or other properties of the protein. But people have also taken these to try to generate new sequences. But this is equivalent to learning one step of the ordering house of diffusion process. All right, so it learns every order, but only one step, one transition really. Or to be more precise, it learns all the transitions after whatever percent of things you sampled. And the advantage of training on every step is that here you see on the x-axis I put how much of the protein you have masked, and the y-axis perplexity, which means the lower is better. Lower means I put more probability mass of the correct amino acid. You can see that when we only mask a small fraction of the protein, if you train on using mass language model or OADM, they do about the same. In fact, the mass language model does a little bit better because it focuses all the model capacity there. But as you increase the fraction of the protein that's masked, you see that the mass language model, which never sees that during training, quickly blows up, quickly makes very arbitrarily bad predictions. So EvoDiff enables a controllable generation of plausible and diverse proteins. Quick reminder, we call it EvoDiff because we're training on many organisms and many functions and many sequences. We ended up settling on this masked diffusion framework. And so we want to evaluate whether individual samples are plausible proteins. We also want to evaluate whether our samples are diverse or if they cover as much of the natural sequence space as possible, because you can imagine get very plausible samples by collapsing to an easy part of the data distribution. And then we want to try to do some conditional generation. So let's start with the plausibility of individual sequences. So one way we can evaluate this is we generate sequences. We use a structure predictor to predict the structure. Then we use an inverse folding method to predict, given that structure, the backbone structure, again, what's an amino acid sequence that's likely to fold to it, and compare that to the original generated sequence. And that gives you what we call cell consistency perplexity, or SC perplexity. We can also look at the sequence PLDT from the structure predictor, which is how confident is that predictor in the structure came out, which is to some extent a measure of how similar is this to structured proteins it's seen before. So we look at the test set, these are natural proteins. We see a range for both the perplexity, the cell consistency perplexity, and the sequence PLDTs, the prediction confidence, amongst the natural proteins. And this makes sense because not every protein, the model isn't perfect, but also because proteins also have what are called intrinsically disordered regions, which are regions that that exist in an ensemble of structures and are not well characterized by a single fit structure. And if we overlay our generations on top, you see that we do manage to come up with some examples of high confidence and low complexity sequences. Although in general, the sequences we come up with do seem to be a little bit less plausible than the natural ones. We can also look at individual generations. These are very cherry-picked to be high confidence, low complexity, and for the most part, pretty different from anything in the training set. Except for the middle one, which is almost identical to something in the training set. but that one will pretty easily put it in there. We can also compare to those other baseline models that I'm claiming our model generalizes. So here's the same plot from before. If we look at the left and right autoregressive model, it actually does a little bit better possibility. So that makes sense in the sense that this is an easier task. The model has to do less work. And these are both trained in a fully generative manner. We can also look at these protein mass language models. Here's DSM-2 at the same size. This is widely considered to be the best protein mass language model at the moment. And if we generate using this from scratch, it's kind of an out of training distribution task. You can see that we actually don't get very many possible sequences at all. All right, so we covered the plausibility of individual sequences. Now we want to talk about how diverse are these? Do we cover the entire natural distribution? All right, so one way to do that. There we go. Once again, we can take any set of sequences. In this case, the test sequences. We run it through a different protein language model. This is a different, it's like ESM2. It's a protein mass language model. But in this case, what the authors previously found is that if you take the embeddings from this model, you can actually use that to build a function predictor. So things that are close together in function space have similar function annotations in the database. You get these embeddings. You can visualize these, and the shape doesn't really matter. They exist. You can also do this for any set of generated sequences, compute the embeddings, and the embeddings should capture something about both structure and function. And then overlay the visualization on top of the natural ones. And the ideal here would be to cover all of these, right? And see here, we do a pretty good job except for that lower left-hand corner where for some reason we have trouble generating. We can also do this quantitatively by looking at a Fréchet distance. So in imaging, people use what's called a Fréchet inception distance. We use a Fréchet 45 distance, and it's looking at how similar one set of embeddings is to the other. So lower is better there. If we compare to those same baselines, we see that once again, the autoregressive model, learning easier task, can give you a slightly lower FBD. The advantages of the diffusion model will be clear when we talk about conditional generation. If we look at the protein mass language model, this actually does very poorly. And that makes sense because generating from scratch is an out of distribution task for that model. And interestingly, we can also compare to the state-of-the-art structured diffusion model, which is a model called RF diffusion. You can see that because the structured diffusion model is trained on a very small and very biased subset of the proteins that are available in nature, it also does not do well on this metric. So we've looked at plausibility, we've looked at diversity, now let's look at the conditional generations. So this is just one simple way to condition these models. And we'll talk in the future work section about some other ideas. But first, in traditional protein design, there's this observation that many functions are mediated by what's called a functional motif, stabilized by a scaffold. So here we're showing the motif in purple and the scaffold in gray. And some examples of this are epitope presentation, which is T cells and T cell receptors and antibodies. There's the part of the MHC or the part of the antibody that actually binds the epitope, and then the rest of it is just kind of hold that part in place. When we look at these viral receptor traps, do the same thing. There's the part that actually binds the virus, and there's the rest of the protein that exists to hold that binding motif in place. We can think about enzyme active sites. And same thing, there's 3 or 4 or 1 catalytic residues that actually help catalyze the reaction. There's some parts that hold the reactants in place, and then there's a scaffold that holds all of those pieces in the right 3D conformation. We can look at protein-protein interactions, which is just where any two proteins, the general term for when any two proteins bind to each other. and perform some function in that way. And there's a binding interface between the two proteins, and then there's, you can think of the rest of the proteins as scaffold for that interface. And the idea here is that one of the applications of models like RF diffusion and volume diffusion is if we're designing in structure space, you can say, this is the structural motif I want in 3D space. Give me a set of residues that will hold that motif in place, right? So you generate the scaffold in structure space, then you design the whole thing in sequence space given that structure. And we want to ask, can we do this task entirely in sequence space without feeding structure directly to the diffusion model? Of course, if I'm showing you this question on a slide, the answer is going to be yes. So the way we do this is we look at that functional motif in sequence space, we fix it, and then we mask everything else and sample around it. In practice, we also sample the distance between the motif sequences and also how much before and how much after. And so here you can see a predictive structure for this generation where the green is that motif, and visually at least seems to match the design motif or the natural motif, but the generated structure, the generated scaffold is pretty different. And we did this without using any structure at generation. It's not, you could be stricter and say we use the structure to pick out which residues to condition, which is true. So we do this with sequence only. You can see some cherry-picked examples here where we show the natural protein, and then we generate a new scaffold for the motif. This is what I just showed. And we can also do this with an MSA. So you also train versions of ELIDIF that generate what's called multiple sequence alignment. And the way that works is you go and take your sequence, you go look in the database for evolutionarily related sequences, you align them so that the related positions are in the same columns. You insert gaps to make that the case. And then we train our diffusion model the same way. We just take that entire alignment and feed it in to the diffusion model. And at generation time, we actually keep all the related sequences and regenerate the query sequence. So we can do the same kind of scaffolding. Here you can see this one's cherry-picked both because the motif RMSD, which is how different the motif is after scaffolding versus the original, is very low. But also because the motif looks impressive in that it's just kind of like three things filled in space. And in this case, the scaffold is also very different. We still have that motif in the middle, but all the loops around it look very different from before. So we can do this at scale. The RF diffusion paper came up with 17 what they call problems, which are these functional motifs that you can create a scaffold. And we define success as your motif RMSD is less than 1 angstrom before and after the scaffolding. So RF diffusion correctly gets 13 of these, or it finds at least one successful scaffold out of 100 attempts and 13 out of the 17. Even with MSA, also there's 13 out of 17, and without using the sequence alignment, the single sequence model gets 8 out of 17. And so here, we're going to dive into these problems a little bit more. Here we're comparing the number of successes for each problem. And the problems are defined by these 4-letter codes, which are the accession codes for each molecule in the structure database. And so for each scaffolding problem, we're showing here whether ELIDIF MSA or ELIDIF CPLUS has more successes. So for most cases, ELIDIF MSA is going to be better because passing all these evolutionarily related sequences gives a lot of information, with a few exceptions. We also compare ELIDIF MSA to RF diffusion. And here we see that we know that they have 13 solved problems each. But we see that there's basically no correlation on whether they're successful. They seem to have orthogonal strengths here. There's also this one PRW, which is interesting in that neither the MSA nor RF diffusion does super well on that, but the sequence one seems to do very well. This one, even the seq does okay, and everything else is terrible at it. We find that there's not many related sequences or homologs in the databases for this, meaning that the structure prediction models are probably struggling with it to some extent. And also, there's the MSA quality is just not very good for evotive MSA. We can also look at, once we have successful scaffolds, how different are those from the original native scaffold? So high means that the generated and the original scaffold are very similar. Low means that they're very different. And you can see that the sequence one is usually more novel, and that makes sense because we're not giving the model a bunch of information about related sequences. All right, so to summarize, we have a model called, really a suite of models called evotive. And what these models aim to do is perform controllable protein sequence diffusion. We call them evolutionary diffusion because we train on evolutionary sequence space. We chose sequence space because, in part, because it's much bigger than structure space. It's easy to get sequences. You go and sequence the DNA of random organisms, you infer what the coding regions are, and that tells you what the sequence is. To get a structure, you have to make it in the lab. You have to crystallize it, you have to solve the structure. So there's hundreds of thousands of structures in the PDB. There's tens of millions of high quality non-redundant sequences you can easily download. If you care a little bit less about quality, you can get tens of billions. The idea here is that in nature, we can observe natural sequences that fold to three-dimensional structures, and that structure mediates some cellular function. We want to look at that data and train a model, in this case evotive, to generate new sequences that hopefully give us new functions. We show that we can do unconditional generation, where you start from all masks, you pick a random decoding order, and you sample one section at a time until you have a full sequence. And in many cases, unconditional generations yield things that look realistic, in the sense that the structure prediction algorithms think they will fold to something that looks like a protein. We also look at motif scaffolding, where you fix a functional motif, and without explicitly giving the model structure, you try to generate a scaffold around it that will hold that motif in the correct position in three-dimensional space to perform the function you want. This is kind of a central thing in structure-based protein design. We just thought it'd be cool to do this in sequence space only. And now we're looking at, amongst other things, we do more broadly do controllable generation, meaning you fix a function, whether that's I want this protein to be super, super stable, I want it to fold to the structure, or I want to catalyze this reaction, or I want to take this protein to catalyze this reaction, but make the other product instead of the one it currently prefers. There's lots of possibilities here. I want to pass that into the model and condition the model to generate sequences that do that. So this is analogous to if you go and play with like stable diffusion or the new one, I think it's called Sora, those videos. Those don't just generate images or videos unconditionally. They take in text that describes the picture of the video. And based on that text, it generates something that hopefully matches it. My personal opinion is that text is not the right modality for specifying biological function, but we want an analogy to that. You can look at the preprint. The code is also available on GitHub, so you can download the model and generate things and play with it. This work was done as part of the BioML group at MSR New England. I want to especially thank the first two there, who are Ava Amini and Alex Liu, who worked closely with me on all this protein and biomolecular generation work. And then below them is Nicola Truzzi, who manages the group and kind of enables us to do all this really cool work. I also want to thank Sarah, who is on the bottom row, second to the right, who is the first author in the paper, who did probably about 75% of the hands-on coding, with me and the intern Nithya doing the other 25%. So here's our summary slide again, and I'll take questions now as well.
My name is
Right, so the reason I don't think text is the right modality here is because it's not precise about the things we care about in a like short, concise way, right? For example, it's very easy to specify the reaction one in some sort of chemistry specific format, like whether that's file strings or something more sophisticated, right? Whereas to describe, if I describe those with the molecule names, that means your model has to learn that. It also has to learn what those molecule names mean and map them to probably these atomic structures of those molecules. And that's just a lot more capability your model has to have.
So are you going to use like some kind of like geometry as a modality? Like some kind of image?
Geometry like of the structure?
Of like cancer cell being destroyed.
Yeah, so the problem there is that's also, I think, a little bit not super well defined, right? There is some work on, for example, you take these cell images of, say, cancer cells or something, right? And then you perturb them by adding other molecules, which can be small molecules, which can be proteins. And the idea is you want to kill the cancer cells and not kill the normal cells, right? And you can gather that from the images. So you could think of a task where it's like, I want the after of adding this molecule to the cells to look like this picture and not this picture. So that's one possibility. Another one in the like geometry idea is, I think people are pretty good now at giving a fixed backbone, designing a sequence of a filter, right? But in real life, proteins are wiggly, which in physics terms means they exist in a Boltzmann ensemble of structures. Will be to specify the Boltzmann ensemble you want, which there's also a problem of like, how do you know the Boltzmann ensemble you want? But one step at a time. Exactly, you can specify the Hamiltonian and say, generate a sequence that will give me a backbone structures in this Hamiltonian. Back there?
Can you say a little bit more about the first state and it's like that surprising failure and I understand expected failure or somehow that efficiency.
Well, which failure modes were surprising to you?
I would have thought that if you distribution that it should just work, that it's fusion should just work a couple of spaces. Like in that case, add nothing.
Right, so there's a few things there. One is that's a UMAP, right? And UMAPs can be misleading. What we actually need to do that we still haven't gotten around to is figure out what sequences are in that zone and why we don't make them, right? And what kind of sequences they are. And whether that visualization is an artifact of like the sampling we did to make it because we didn't want to do to UMAP 10 million things, or if it's an artifact of the UMAP, or if it's something real and what is that real thing. The other part is that I don't show this, but due to compute constraints, our model is actually very undertrained. So we could probably do better if they just gave us like another month of compute. For reference, it's 640 million parameters. We train about 40 million sequences, and it's trained on 32 V100s for about 4 weeks.
And that's not enough.
For comparison, ESM2, which is the same size mass language model, was trained on either 256 or 512 V100s.
like a CNN.
We had a stable one that worked. Also, unless you get longer context for the same compute cost, right? Because your transformer scale is sequence squared.
Yeah.
Yeah, so even with B100s, you can easily do like 1024, but to get to 2048 with that size of transformer would take some tricks. Not to say you can't do it, but
So definitely Jack and or
yes. Yeah, it's a massive likelihood. Well, to be, it's, in practice, yes, the original paper justifies it with, as like maximizing the posterior, but in practice, it's trained maximum likelihood of the original sequence.
So it seems that the potential of this scaffolding problem is continuous small segments. There is some hidden assumption that their structure is not dependent on the rest of the sequence. So like why after you mask all the rest of the sequence, would you get the same structure, relative structure between the small motifs which are unrelated?
I mean, you don't, right? That's why the success rate is like 20 out of 100, not 100 out of 100. But the question is, do you get some? Right, because we're not actually specifying directly the 3D coordinates the way you would in a structured diffusion model. That problem in general is kind of a toy problem in the sense that even in the structure space, just being able to scout, it's not that it's not necessarily, it's that the kinds of functions you can design this way are pretty limited. So binding is the obvious one. You can maybe do some simple enzymes, but beyond that, anything that's not relying on a single very stable static structure is very hard to design this way.
I want to ask you a question on the diversity of your data set, right? I mean, you show this UMAP and there's some region that nobody seems to be able to predict, right? And I'm just wondering if there's just a implicit bias coming from this.
Right, so our data set is deduplicated in the sense that people cluster to 50% identity and you pick the cluster representative for every cluster, right? However, the way they pick the cluster representatives definitely probably biases things a little bit. I think it's like there's some preference for longer sequences and there's some preference for sequences that are better characterized, which usually means sequences from model organisms. And but that's why we need to look at those and see if is it things that are unstructured, is it things from that are more unrelated to other sequences, is it sequences where the cluster representative is from viruses, right? The viral proteins that are harder to do, or is it archaeal proteins? I doubt it would be like E. coli proteins or human proteins.
But it's great things you're still doing.
Right, yeah, but I suspect that the biases in the training set are at least partially responsible. Because it's a combination of what things are easy to learn and what things are underrepresented.
Any other questions? All right, let's thank the speaker again.